Namaskar friend, welcome to the channel Pharmacy Dictionary. In today's topic we are going to study about the next part of pharmacokinetic, that is transport of drug across the cell membrane. Have you seen any kind of barrier on the road? Have you? There are two types of basic barrier. First one is considered as a toll tax and the second one is known as the mineral checking barrier. In this today's topic we are considering the example of this mineral checking barrier. Some truck or some goods vehicle is going on and suddenly there is a barrier. The barrier pulled down to just check whether it is having any kind of unauthorized or illegal material or minerals inside the truck or any kind of vehicles. Then they took time. After complete checking that is it okay, then they will allow it to go further. If something is wrong, they will stop they will take that truck or any vehicle in their custody and the further study will be going on. After confirmation that nothing is wrong, they will also allow them, and if they found that is illegal, they may arrest them. So what is the logic behind this example and today's topic? Today's topic we are going to study about the different process or different transport process across the cell membrane when the drug is taken, when the drug is administered, when the drug is absorbed in the blood vessels. So how the blood which is dissolved in the blood moves through different parts of our body. Can any kind of drug move all across our body, or there is some specification like lipid solubility and the water solubility having difference for their distribution point of view? Yes, all this question is answered in yes. Everything is having any specificity, and that is a regulation process in the transport across. So what are the basic transport process across the cell membrane? There are mainly passive transport, carrier-mediated transport, and the third one is endocytosis or exocytosis way of transport. And under these headings there are subheadings too. So if you take the example of passive transport, there are subclasses like simple diffusion and the second one is filtration. So I will explain each and every point one by one in the whiteboard section. The second one is carrier mediated, that is active transport or facilitated transport. The third one is endocytosis and exocytosis having subtype of pinocytosis and phagocytosis. So let me explain you the basic meaning of these three type of transport system and their subtype along with their respective example. So first one is passive diffusion, or we can say passive transport. What is the meaning of passive? To understand the meaning of passive, you should know the meaning of active. What is the meaning of active? Whenever you are in active condition, you are in alert condition, you need to produce energy or you need to apply that energy into any work you are assigned or any work you are intended to do. That is called active. I am sleeping, I am not doing any kind of activity, but if I have to pull anything or I have to throw anything, then I have to apply the energy. So this is called active. But what is inactive or what is passive? Anything which took place without applying any kind of energy. If you are throwing something from bottom to the top, you will need to apply the energy. But if you are dropping something from the top to bottom, you need not to apply any kind of energy. Just you have to leave that object so that that will come automatically according to the gravitational force. So there are basic difference between active and passive. So passive transport means any kind of transport which does not require energy for the facilitation or for the driving of any activity. And in this current whiteboard section, I will explain this passive transport. Then like in next board I will explain you another part. So these are simple diffusion and filtration. Simple diffusion as such I have explained you in this channel. The process of diffusion is a movement of molecule from the region of higher concentration towards the lower concentration without need of any energy, without need of any facilitation force. That is called simple diffusion. And what is the meaning of filtration? As you have performed this filtration process so many times in your usual or daily life, that is same can be applied here. In case of filtration, have you filtered the water with the help of clothes, muslin cloth? Have you filtered the tea while making a tea, or have you filtered any kind of object from the water itself? That is simply known as a filtration, and the process or principle of filtration is simply based on its particle size and the availability of pores of any filter aids. Like filter paper, only those substances will be allowed to filter which is having a smaller size than the pores of the filter paper. The higher size or higher substance will not be allowed to go through the water. That is the simple principle behind the filtration. So the next one is carrier-mediated. As its name indicates, to facilitate the transport process, we need some carrier. Have you seen the carrier in a bicycle? Yes, if you have seen this carrier in your bicycle, then you can understand what is the work or what is the assigned task of the carrier. In carrier, we used to carry or we used to place the college book, college bag, school book, school bag, etc., etc. Though carrier means the substance which is being carried out by the help of some object, that's called carrier mediated. Suppose I am superstar, well-known superstar, Hollywood star, Bollywood star, so I can enter in any kind of Bollywood event, but you or someone else, just for example, I'm not expecting that is real thing, so someone is not allowed to go inside the party hall or anything, so they will be stopped by the bodyguard. So what can they do to enter in the classes? If I know them, I will come out, I will take them with myself, and then I can move throughout the party or anywhere in the party, so they will not be hindered process. No one be going to stop that particular person because he is with me. And same process can be applied with this kind of process. Any drug molecule which is not allowed to move in a alone phase can be taken help by any kind of carrier, that is suitable carrier, to move across the membrane. So this is called carrier mediated, and there are two subtypes of carrier mediated transport across the cell membrane. These are active transport. Now there is a requirement of energy because it is a process that is not able for the person or particle or drug which is single alone, which is a single particle cannot be moved through. Second one is facilitated, that need facilitation, that means need support from others, like as name indicate. Now third one is endocytosis and exocytosis. This is not very common, but this is the important phenomenon of transport of drug across the cell membrane, even for different types of unwanted material too. Endocytosis and exocytosis of two words. Endo means inside the cell and exo means outside the cell. So anything which is coming inside the cell is considered as an endocytosis, and which is going out or which is being thrown by a body or cell, that is called exocytosis. And depending upon the nature of material, state of material, this can be classified into two types. The first one is pinocyte, the second one is phagocyte. As its name indicates, pino is representing the liquid condition. In Hindi, pino, that means to drink something. That's pino is for drink something, and we drink only the liquid material. So in this case of pinocytosis, the substance which is being engulfed or which is being excreted should be in a liquid state. And the second one is phago, that's converted from the phagia, that means to eat, and we eat solid materials. That is why it is called phagocytosis. In this process, any cell engulf the solid particle of drug or excrete, because exocytosis expel any kind of drug or any kind of substance which is in solid state. So these are some classes and their subclasses. Now coming to the simple diffusion specifically, because we have to understand all this process one by one. So in this whiteboard section, let me explain you the simple diffusion first, then we will go for the filtration in the same video with the next whiteboard section. So in simple diffusion, the points has been written as driving force is a concentration gradient. What is the meaning of driving force? What is the meaning of driving itself? Driving a person who is driving the vehicle, car, motorcycle, or whatever it is, in a right path, in a right direction, with applying his knowledge, his energy, or whatever the requirement are there for the driving purpose, he is applying all things. So any person who is driving is called driver. And the driving force here, what is the driving force? In case of vehicle, the driving force is throttle, is a powerful machine that run the chain and run the wheels across. That is called driving force. But in this case of simple diffusion, the driving force is not energy, it is concentration gradient. Now what is the meaning of concentration gradient? That means difference between the concentration of two particles or two drug or two any circumstances, as we can say in a broad sense. So if there is a difference of concentration between two particles or two substance objective, comparatively this is called concentration gradient. Suppose I have two glass. In one glass I have placed 10 spoon of sugar, and the second one I have placed just only 1 or half tablespoon of sugar. So what is the difference between these two glass? Level of water is same, but the difference is in sugar concentration. We can say amount of sugar, you can also say after dissolution the level of sugar, dissolution of sugar, concentration of sugar is different between these two types of glasses. Now coming to the second important points, I have included only points here. So drugs move from the higher concentration to an area of a lower concentration, as I have given you the example. Only the movement of particle or substance will be from higher concentration to lower concentration. That means from the higher concentration of any particle will move towards the lower concentration. That is the main important point of this simple diffusion process. Now third point, does not involve, this is important, does not involve carrier, because it is independent of energy, because it can move alone across the membrane with themselves without need of any energy. That is why they do not need or they involve any kind of carrier or substance which will help them to move through. The next is not saturable. This process will not be saturable until the concentration is maintained. Low structural specificity, there is not such important, but I can say the 0%. There is no, in percentage, no need of structural specificity. Any molecule which is having difference between concentration gradient can follow this rule or can follow this process. Next one is speed of diffusion depends on the concentration gradient of their lipid solubility. This is very important. You should remember this, because in case of GPAT, any other sessional examination or any other type of competitive examination, such questions can be asked. So see, speed of diffusion from movement of one part to the other part, what is the speed? So this speed, this depends on many factors. The factor are concentration gradient, as I have said, this is higher concentration, this is lower concentration, and their lipid solubility, whether they are soluble in lipid or not. This is a very important phenomena. I will going to explain you in a such diagrammatic way soon. So this depends upon the lipid solubility. Lipid soluble drug and different types of other parameters, lipid solubility and lipid soluble drug, unionized drug. What is the meaning of unionized drug? The drug should not be in ion form. That is called unionized form. This is very important. I will explain you this in case of distribution under the pharmacokinetic profile. So all these points increase the rapid movement of drug across the cell membrane, across the simple diffusion process. In the simple diffusion process, I must say, so most of the drug follow this kind of phenomena. Many of the drug which we have taken or we are going to take in the future time will follow the same process of movement. Now I should explain you with the help of some diagram. In this case, I am going to draw a cell membrane which is made of phospholipid bilayer. So this is the head portion, this is tail portion which I am going to draw. I did not draw it earlier because I want to explain few points here. So these are the cell membrane and these are space, pores available inside. Now again I have to type the diagram, I have to make the diagram over here. So all these circles like structure are head portion of the phospholipid bilayer and these are tails. So this phospholipid bilayer or cell membrane are made up of different combination of head and tail particles. As you can see, there is a gap between this cell membrane. So we can say this is a extracellular portion or this is intracellular portion, or we can say these are the cytosol inside the cell. So here I have already mentioned the criteria. The green one shows the passive diffusion of water soluble drug through the aqueous pores in the channel. So this is in green color. So assume these are the drugs which are aqueous or water soluble, so they can move across the cell membrane only through the pores available in the membrane. These are the pores, no barrier are present, so they can move only through this way. And in case of red circle, you can say this is the passive diffusion of lipid soluble. Main difference is water soluble and the second one is for lipid soluble drug that will dissolve in the cell membrane and move across. So there is no need to have any kind of pores. So this is a lipid soluble drug, so there is a pore for water soluble drug, but there is not required, there is no need of pores for the lipid soluble drug. They can move across the membrane via dissolving themselves in the membrane. So two important points are very important, and for the next part of the same video, in the next whiteboard section, we are going to study about the filtration process of passive transport. This is a very important. So let's move to the second whiteboard section. Now let's move to the another method of drug movement across the cell membrane. The next process is filtration. You know the exact meaning of filtration? This is a process in which a small molecular substance or small size of molecular substance move towards the pore available on the cell membrane. But if you are talking about the drug, so the drug which is water soluble will go through the cell membrane only with the help of aqueous pore present in the phospholipid bilayer, yani ki cell membrane. So this is called filtration process. We have used so many filtration process in our daily life, like making a tea. Screening the tea is the simplest example of the filtration, like we have to filter the tea leaf and we have to accept that extract of T. So there are so many other examples of filtration too. I am going to explain you with the help of common household utensils. As you can see in this screen, there are so many sizes of a screen. We can say pore size of a screen. For some time, please assume this as a membrane, or we can say that as a phospholipid bilayer made up of membrane. So they are having different size of pores. This is the smallest size. So only those drug, only those agent, now here we can use agent also, those drug or those agent which is having smaller size of particle as compared to the pore size of this screen may only pass through this screen. Otherwise they will not allowed to be passed. And in this case, this is the largest size of a screen. In this size of a screen, many of the molecule as compared to the previous one, like this one, can pass across through this barrier. So this depend upon the size, depend upon the pores available in the cell membrane. It is applicable only for the aqueous or water soluble drug, as like this one and this one. So all these screen are in different size. Only those molecular size of drug, those molecule of the drug which are in smaller size as compared to the pore size can only pass through. This is a repeating word, and please make sure you have to understand this point very clearly. Now see the points. Passage of drug through aqueous pores in the membrane, it is characteristic phenomena of filtration. That means there is the requirement of aqueous pore if you want to allow the drug from the extracellular to the cytosol of the cell. Pore is very important. Which type of pores? Aqueous pore. That means those drugs are water soluble. It is clearly in this point very concisely. Now next see, water soluble drug with the molecular size smaller than the diameter of the pores, which is generally 7 Armstrong, can only cross the membrane, as I have shown you in the diagram. Let me show you another example. Suppose for some time, this is an example of phospholipid bilayer. You can see this is made up of sponge, and we can assume this as a cell membrane. So to pass any drug via the cell membrane, it has two options. First, the drug should be water soluble if the drug is water soluble, and the second, if the drug is lipid soluble. If the drug is lipid soluble, then it may cross across the cell membrane without need of any pores, as like you have seen in this representation. But if the drug is water soluble, it cannot pass as such. It need a aqueous pore, and in that case there should be a pores. Like you can see this, we are removing this kind of space, then it has to be a pore like this one. So if it is pore, then it can easily pass through this. So these are the difference between the aqueous soluble or lipid soluble drug movement across the cell membrane. Pores, aqueous pores is required in case of filtration, and rest of case you have seen what is happening here. Now see the difference between other type of movement. Movement is along the concentration gradient, and this is only possible when the concentration gradient is different. So along with the concentration gradient. Now let's see the example. The example of this filtration is urea in case of urine filtration process. Now another point says the capillaries in the certain tissue like brain and testes lack the aqueous pore, so the drug cannot reach to that particular site. As we have seen in the first introduction topic, there are so many barriers like blood-brain barrier, blood-testis barrier, and different types of blood-placental barrier. Out of which a few examples are brain, that means blood-brain barrier, and testis, that means blood-testis barrier. They do not have any of aqueous pores. So if they do not have aqueous pore, how the drug which are aqueous soluble or water soluble can move through? Because they are lacking the aqueous pore, and that is why those drugs cannot be reached to the site of action if the sites are brain and testis. That is why we have remembered this point. Only lipid soluble or highly lipid soluble drug can cross blood-brain barrier or blood-testis barrier. This is the most important question generally asked in your semester examination. Now coming to the next method of transport, that is carrier-mediated. As the name indicates, it is a movement of drug across the cell membrane with the help of carrier. What is the meaning of, basic meaning of carrier? A person or agent which is used to carry something. If you are talking about the person, it is a person. If you are talking about the goods, carrier, that may be carrying the goods. So carrier means whoever is carrying something, whoever is responsible for transporting something from one place to the another, one part to the another, is simply known as a carrier. So what are the basic responsibilities of carrier-mediated transport system? So first one is active transport. Under the carrier-mediated, first classes is this one. In this, this is the against the concentration gradient need energy. In case of passive diffusion, we have seen that is totally independent upon the energy, that is not required energy. But in case of active, as name indicate, I have also explained you regarding this active word. So this is process which is taking place against the concentration gradient. No need to be as such. They will not follow the basic rule. They will go against the concentration, and to go against the concentration we need energy. So that need energy for the process of active transport system. Now carried out by specific carrier protein. This process of carrier mediated transport required a specific type of carrier protein which will carry the drug, and that will transfer the drug from extracellular to intracellular. They will dissociate and then will release the drug, then they will again go back to the cell surface, and then again will carry the drug molecule and will move to the next process. So this movement process will continuously taking place. Now next one, only drug related to the natural metabolites are transported by this process. Example are levodopa, iron and amino acid and sugar. What is the meaning of this line? See, in case of metabolism, we are going to study about the metabolism very soon after the coming topics. Then in this case of metabolism, the process converted from complex molecule to simplex molecule. In some case, few drugs are not active as such. That means like Parkinson case, we need to give levodopa, because levodopa is active form, but actual drug is dopamine. We should give dopamine, but dopamine is not active as such. That is why we have to give the prodrug of dopamine, that means levodopa, and levodopa will convert it into dopamine after metabolism. So that dopamine which is converted by the levodopa can only be transferred through this type of active transport in our body. So that is a specific phenomena, or we can say specificity, that natural metabolites can only be transported by this mechanism. Now coming to the other point, on other side of the cell, that means inside the cell we can say, as this is a cell, this is the extracellular and this is the intracellular. So this extracellular is responsible for the conversion of for the binding of drug with the carrier, and they will transport the drug to inside. Now we are talking about this one, intracellular side. On the other side of the cell, complex, that is a drug carrier complex, disassociate. Now this is the drug and this is carrier, just come inside the cell, they will disassociate and they will be separated, and the drug will be another molecule and the carrier will be another molecule. That means there is a differentiation of drug and carrier, or we can say dissociation of drug and the carrier. So what is going to happen on the other side of the cell? Complex molecule dissociate and carrier move back to transport other drug molecules. This is the important, as I can explain you with this help. This is the carrier with the drug, they will come inside, they will dissociate, that carrier will go again to the surface, then again bind to the drug, again come inside, and this process will be going on continuously take place. So this is the important phenomena. I will explain you with the help of diagram in next coming video. Now let's take important points. There is a note, other drug following the same principle may affect the rate of this process, that means carrier mediated transport process. If the another drug which you have taken simultaneously with the same drug which is following the same transport process may affect the rate of transport of each other, they may prolong or they may delay the process of transport. So friend, this was a continuous part of the previous whiteboard section. In this whiteboard section we have studied about the basic types of transport mechanism. In the next whiteboard section we will continue the further remaining parts. Thank you very much for watching this video. If you want to take the screenshot of this whiteboard section, you may have it after some time. And the reference for today's topic is P. Jagdish Prasad and K. D. Tripathi. Thank you again.